We just did slight revision of our earlier class. Now we'll proceed to today's class. Today's class we'll try to understand the oxygen dissociation curve for hemoglobin and myoglobin. You all know that hemoglobin carries oxygen to different tissue cells, but myoglobin stores the oxygen in the muscle and releases in need. That means when, just like the muscles, when deprived of oxygen, it releases the oxygen. Okay. This is the main function of oxygen and myoglobin and hemoglobin, and we'll try to correlate this function with their structure by knowing about the structural functional relationship. And to understand that, we have to know about the oxygen dissociation curve. In the last class also, we have already discussed the hemoglobin can bind only one molecule of oxygen, myoglobin can bind only one molecule of oxygen, whereas hemoglobin can bind 4 molecules of oxygen. Okay. Now we are going to oxygen saturation curves. The degree of saturation of these oxygen binding sites on all myoglobin or hemoglobin molecules can vary between 0, that means all the sites can be empty, or all the sites can be full. A plot of this wide degree of saturation measured at different partial pressures of oxygen is known as oxygen dissociation curve. We'll see and try to explain with the help of this curve, oxygen dissociation curve. That means the degree of saturation of the oxygen binding sites, on all hemoglobin or myoglobin molecules. This is known as oxygen dissociation curve, and the oxygen dissociation curve for hemoglobin and myoglobin so important different characteristics. We'll see that the curves for hemoglobin and myoglobin show different important differences. So this is the curve, oxygen dissociation curve for hemoglobin and myoglobin. Now let's see, this is your x-axis, this is the y-axis. Can you see my slide also?
Yes, sir.
And cursor, are you seeing my cursor? Can you see?
Yes, sir.
Okay, then I will proceed. Okay, we're talking about the oxygen dissociation curve for myoglobin and hemoglobin. Now this is the x-axis. On the x-axis you can see the partial pressures of oxygen, and on the y-axis percentage saturation with oxygen. This is the partial pressure of oxygen, and on the y-axis percentage saturation with oxygen. And you can see two different curves here. One is simply rectangular hyperbola type. One is simply this one. This is rectangular hyperbola type, whereas the other one, this one for hemoglobin, this is the sigmoidal shape. This is a rectangular hyperbola, whereas this is sigmoidal shape. Okay. By seeing this curve, first of all, you see that the curves are different for hemoglobin and myoglobin. The myoglobin, it is having rectangular hyperbola, but myoglobin is having rectangular hyperbola, but this hemoglobin is having the sigmoid curve. Okay. Why they are having the different curves, we'll discuss. Now this is the partial pressure of oxygen on the x-axis. As we increase the partial pressure of oxygen on the x-axis, now the percentage of saturation will also be increased. In this y-axis you can see. In the case of this myoglobin, as you can see, percentage saturation, just like percentage saturation is, you can see 0, 50, 100. 0, 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, in this way it is ranged. Now if you see here, when the percentage saturation of myoglobin is 50%, the partial pressure of oxygen is only around 1 mmHg. That means when the partial pressure of oxygen in the tissues, we'll be talking about the muscles, when the partial pressure of oxygen in the muscles is 1 mmHg, In that condition also, this myoglobin will be 50% saturated with oxygen. Now, if you see this for hemoglobin, this is the 50% saturation, and to get 50% saturation, the partial pressure of oxygen should be around 26 mmHg. Okay, from this x-axis you can see the partial pressure of oxygen, varying partial pressure of oxygen. Now we are talking about the oxygen dissociation curves for hemoglobin and myoglobin. Oxygen dissociation curve for hemoglobin and myoglobin. So different characteristics. You can see for hemoglobin it is sigmoidal shape, whereas for myoglobin it is rectangular hyperbola. It is rectangular hyperbola means there is the simple binding of oxygen with the myoglobin, binding and release. But this sigmoid shape means there is not just binding and releasing, but there are some complex relations in binding and in releasing oxygen also. That means it also explains the cooperativity of binding oxygen with hemoglobin. Now let's see here. If you see here, the oxygen dissociation curve for hemoglobin is steepest. You can see the steep slope is steepest at the oxygen concentration that occur in the tissues. In the tissues means nearly the oxygen partial pressure of oxygen would be 20, 30 or 40. That means in this region, in the region of 20, 30 and 40 region, this curve is steepest. That means the percentage saturation will differ much. That means it will carry the oxygen from the lungs, and that oxygen it can deliver to the tissues, because in the lungs partial pressure of oxygen in the blood is around 100 mmHg. From that it will bind with the hemoglobin and goes to the tissues where the partial pressure of oxygen will be around 20, 30, 40, this much in the range, in this range. And this curve is steepest. That means with the change in slight partial pressure of oxygen also, percentage saturation can change. That means when going to the tissues, this hemoglobin can release the oxygen molecules. But if you see for this myoglobin, you can see even if the partial pressure of oxygen is 1 mmHg, it is already around halfly saturated. Yes, and that means in the partial pressure of in the range of partial pressure of oxygen in the tissues, it will be binding with itself the oxygen it carries. That means it cannot deliver. It can only deliver oxygen only when the partial pressure of oxygen is very less. As you can see, the steep slope towards 1, 2, 3, 4, 5, around this 5 to 10. You can see here, this is 40, isn't it? 10, 20, 30, 40. That means this steep slope is within the 10. That means if the partial pressure of oxygen is let's say more than 10, approximate more than 10, now that means it has the high affinity to bind with the oxygen. It has the high affinity to bind with the oxygen means it cannot deliver oxygen to the tissues, which partial pressure is nearly 20, 30 or 40 mmHg. In this partial pressure, it is not able to release the oxygen. It will only be able to release oxygen in the low partial pressures of oxygen, which occurs in the exercising muscles. In that condition, it will release the oxygen and utilized by the muscles. That's why this myoglobin is suited for the storage of oxygen, whereas this hemoglobin is suited for the carrying of oxygen to the tissues. If myoglobin would be the molecule to carry oxygen from the lungs to the different tissues, what it would do? It would always carry the hemoglobin. It would always carry the oxygen along with it, because it takes around less than 10 mmHg, less than 10 mmHg of partial pressure of oxygen, so that the myoglobin will release the bound oxygen. But normally in the tissues, that is not the partial pressure of oxygen, and this much less partial pressure of oxygen would only occur in the exercising muscles and all. If this myoglobin would carry oxygen from the lungs to different parts of the body, then what would happen? It would be carrying and carrying. It will not be able to release the oxygen to the tissues. because to release the oxygen in the tissue, what should be there? There should be very low partial pressure of oxygen. That is not the case in our body. That's why this the curve of hemoglobin, this has the steep slope in the region of partial pressure of oxygen in the tissues. You can see 20, 30 or less than 40. In this region it has the steepest slope. Steepest slope means even if there is a slight change in partial pressure, it can release the oxygen. That means steep slope here means percentage saturation will be changed. Percentage saturation will be changed means the oxygen it has carried from the lungs would be released in the tissue. That's why this hemoglobin is suited as a or suitable molecule to carry oxygen from the lungs to the different tissues, whereas this myoglobin is suitable to store the oxygen in the muscles, which would be released only during the times of need. That means when the partial pressure of oxygen in that tissue, in that muscle is very less than 10 ml. In this region also it will release some, but up to this is the steepest part, you can see, isn't it? Up to 10. Okay, this is about the oxygen dissociation curve for hemoglobin and myoglobin. Oxygen dissociation curve for hemoglobin and myoglobin in the x-axis, partial pressure of oxygen; in the y-axis, percentage saturation with oxygen. You can see as the partial pressure increases, this percentage saturation will also increase in both the cases, but the increase in the percentage saturation will be very much higher in the very less partial pressure of oxygen in case of myoglobin. That means in the less partial pressure of oxygen, this myoglobin would bind the oxygen along with itself. But in the case of hemoglobin, as you can see, there is a slight change, and in this region, that means the region which is the partial pressure of oxygen in the tissue is at the steepest slope. That means in that region, even small change the partial pressure would occur there, when percentage saturation will be changed. How the percentage saturation will be changed? By releasing the oxygen. That means the tissues will be able to get oxygen. Okay. This is the oxygen dissociation curve for hemoglobin and myoglobin. This is the most important slide. However, I have written in words also the same thing in the other slides. I will go through the words also and to make you better understand about the setting. Myoglobin. The oxygen dissociation curve for myoglobin has a hyperbolic shape. This reflects the fact that myoglobin reversibly binds a single molecule of oxygen. That means there is no complex. Thus, oxygenated myoglobin and deoxygenated myoglobin exist in simple equilibrium, as you can show myoglobin plus oxygen, oxygenated myoglobin. The equilibrium is shifted to the right or to the left as the oxygen is added or removed from the system. Myoglobin is designed to bind oxygen released by hemoglobin at low partial pressure of oxygen found in the muscle. Myoglobin in turn releases oxygen within the muscle cell in response to oxygen demand. That means when the concentration of oxygen is very low, that means the partial pressure less than 10 mm Hg. Hemoglobin, the oxygen dissociation curve for hemoglobin is sigmoidal in shape, indicating that the subunits cooperate in binding. We know that the hemoglobin has 4 polypeptide chains. This sigmoid shape of oxygen dissociation curve also explains in the initial times. um, that means with the change in partial pressure of oxygen, there is not much saturation with oxygen. But with later on, there is a steep hill. This steep hill is due to the cooperative binding of oxygen. Cooperative binding of oxygen by the 4 subunits of hemoglobin means that the binding of the oxygen molecule at one heme group increases the oxygen affinity for the remaining heme groups in the same molecule. That means for the first, we know that hemoglobin consists of 4 polypeptide chains, and 4 oxygen molecule will be bind to the hemoglobin. And for the first oxygen molecule to bind to the hemoglobin, it is very difficult, not that easy. But for the second, it is easier, for third, more easier, and for fourth, the easiest. This is known as the cooperative binding. Cooperative binding of oxygen by the 4 subunits of hemoglobin means that the binding of oxygen molecule at one heme group increases the oxygen affinity of the remaining heme groups in the same molecule. Although it is more difficult for the first oxygen molecule to bind to hemoglobin, the subsequent binding of oxygen occurs with high affinity, as shown by the steep upward curve in the region near 20 to 30 mm of Hg, as we have seen with the above graph. Okay, this is the hemoglobin binds oxygen with increasing affinity, because this is the hemoglobin representation of the hemoglobin. 4 polypeptide chains are shown, and in the 4 polypeptide chains there are 4 heme groups, and on each of the heme group oxygen would be bind. And for binding of this first oxygen, it is difficult, but for second it is more easier, even more easier, and the easiest. This is showing the increasing affinity for oxygen. Because of this increasing affinity, the second or the third oxygen can bind easily with the hemoglobin molecule. That's why there is a stiff yield in that region, as you can see. Now let's see the significance of the sigmoidal oxygen dissociation curve. What is the significance of that? The steep slope of the oxygen dissociation curve over the range of oxygen concentration that occur between the lungs and the tissues permits hemoglobin to carry and deliver oxygen efficiently from sites of high concentration to the sites of low concentration. That means the high partial pressure of oxygen which occur in the lungs to the low partial pressure of oxygen which occur in the tissues. A molecule with a hyperbolic oxygen dissociation curve, as in the case of myoglobin, would not achieve the same degree of oxygen release within the same range of partial pressure of oxygen. Instead, it would have maximum affinity for the oxygen throughout this oxygen pressure range, the partial pressure range, that is the representation of the concentration of oxygen actually, throughout this oxygen pressure range, and therefore would deliver no oxygen to the tissue. That means if hemoglobin would be present in the red blood cells instead of myoglobin, then what would happen? That hemoglobin will carry, myoglobin would carry the oxygen, but it would not be able to deliver the oxygen in the tissues, where the partial pressure is in the range of 20 to 30 mmHg. Because at 20 to 30 mmHg, because in the range of 20 to 30 mmHg, it is already almost fully saturated. Fully saturated means now it will not release. Fully saturated means in this condition also it has high affinity with the oxygen. That means if it is having the high affinity with the oxygen, that means it will not release the oxygen. Okay. This is about the oxygen dissociation curve for hemoglobin and myoglobin. Now let's see about the allosteric effects. The ability of the hemoglobin reversibly, that means to understand more about the sigmoid shape of uh, oxygen dissociation curve for hemoglobin, we have to know about the allosteric effects or the allosteric effectors. The ability of hemoglobin to reversibly bind with oxygen is affected by the allosteric effectors. That means the allosteric means, allo means different, steric means side. By binding at a different side, they will increase or decrease the affinity of the hemoglobin molecule to bind with the oxygen molecule. The ability of the hemoglobin to reversibly bind oxygen is affected by partial pressure of oxygen, that is the most important, pH, partial pressure of carbon dioxide, and the ability of 2,3-bisphosphoglycerate. About this 2,3-bisphosphoglycerate, you might be just thinking that, oh, where is this molecule coming from? This 1,3-bisphosphoglycerate is the intermediate in the glycolysis, and that 1,3-bisphosphoglycerate would be converted to 2,3-bisphosphoglycerate in a cycle known as Rapaport-Luberin cycle. That you will be studying while studying about the glycolysis, Rapaport-Luberin cycle, and in that Rapaport-Luberin cycle, 2,3-bisphosphoglycerate would be formed from 1,3-bisphosphoglycerate in the normal pathway, which we have seen in case of glycolysis. Okay, allosteric effectors are partial pressure of oxygen, pH, partial pressure of carbon dioxide, the availability of 2,3-bisphosphoglycerate. pH, that means it is related with the hydrogen ion concentration. If more hydrogen ion concentration means it will release oxygen. Partial pressure of oxygen also ultimately related, carbon dioxide. Carbon dioxide related to the carbonic acid. Carbonic acid in our body related to the hydrogen ion. That means more hydrogen ions, then it will release the oxygen. That's why hemoglobin is able to release the oxygen in the tissues, because in the tissues the pH is slightly low due to the production of carbon dioxide due to the metabolism. And next is the availability of 2,3-bisphosphoglycerate. This 2,3-bisphosphoglycerate also helps to release the oxygen from the hemoglobin molecule. We'll see them one by one also now. These are collectively called allosteric effectors because their interaction at one side of the hemoglobin molecule affects the binding of oxygen to the hem group at other locations of the molecule. The binding of oxygen to myoglobin is not influenced by allosteric effectors because myoglobin, it is a single chain, and its characteristics is also not that complex, as we have seen in oxygen dissociation curve. Simple hyperbola curve it has shown. That means there is no effect of the allosteric effectors. No problem, effectors. And now actually to understand about the allosteric effectors, we have to know about the heme interaction. That means in the hemoglobin molecule there are 4 heme, and this heme interactions also increases the cooperativity. That means this, due to the heme interaction, there is the cooperative binding. That means after one molecule of oxygen binds to the one heme group of one polypeptide chain, now there will be slightly change in the position of the other polypeptide chains due to this heme interaction, and due to the slight change in position, then it will be easier for the subsequent oxygen molecules to bind. This we have to study in heme interactions. The sigmoidal oxygen binding curve reflects specific structural changes that are initiated at one heme group and transmitted to other heme groups in the hemoglobin tetramer. That means once one oxygen molecule binds to one heme of one polypeptide chain, there will be the heme interaction with the other heme groups of other polypeptide chains within the hemoglobin molecule. That slightly changes their position, and that makes the second, third, and the fourth oxygen to bind more easily. The net effect is that the affinity for hemoglobin for the last oxygen bound is approximately 300 times greater than its affinity for binding with the first oxygen. Okay, we are talking about the cooperativity in binding or the cooperative binding. That means for the first oxygen it is difficult to bind, but easier for the subsequent oxygen to bind. Now let's see. Oxygen, hemoglobin takes oxygen from the lungs and it delivers oxygen to the tissues, isn't it? This is about the loading and unloading of oxygen. Let's try to understand this in the simple way from this slide first. Cooperative binding of oxygen allows hemoglobin to deliver more oxygen to the tissues in response to relatively small changes in partial pressure of oxygen. As you have seen, there was a steep hill in the region of partial pressure of oxygen in the tissues. For example, in the lung, the concentration of oxygen is high and hemoglobin becomes virtually saturated with oxygen. In contrast, in the peripheral tissues, hemoglobin releases much of its oxygen for use in the oxidative metabolism of the tissues. Okay. Now this loading and unloading of oxygen is better explained by the Bohr effect. This Bohr effect is also important while studying about oxygen and while studying about hemoglobin and myoglobin and oxygen dissociation curve. This Bohr effect explains how the oxygen is delivered. The binding of the oxygen to hemoglobin decreases. The binding of oxygen to hemoglobin decreases with increasing hydrogen ion concentration, or when the hemoglobin is exposed to increased partial pressure of carbon dioxide. Actually, partial pressure of carbon dioxide means ultimately it will be going to form H2CO3, and from that H2CO3 hydrogen will be formed. That means when hydrogen is there, the binding of oxygen to hemoglobin decreases with the increasing hydrogen ion concentration, because when there is the increasing hydrogen ion concentration, for example in the tissues, what would happen? That hydrogen ions will bind with the hemoglobin molecule, the charge groups of the hemoglobin molecule, and it will form a salt bridge, and that salt bridge will stabilize the cloud form, and in this process oxygen will be released, and tissues will be able to get the oxygen which is carried by the hemoglobin. Just the opposite thing happens in the case of lungs. The binding of oxygen to hemoglobin decreases with increasing hydrogen ion concentration. Increasing hydrogen ion concentration is found in the tissues. Or when the hemoglobin is exposed to increased partial pressure of carbon dioxide, this phenomenon is known as the Bohr effect. Bohr effect means the affinity of oxygen binding with the hemoglobin decreases with the increasing hydrogen ion concentration. This is known as the Bohr effect. And due to this Bohr effect, hemoglobin is able to release the oxygen in the tissues. Bohr effect is primarily responsible for the release of oxygen from oxyhemoglobin to the tissues. This is because of increased partial pressure of carbon dioxide and decreased pH in the actively metabolizing cells. Now let's see the same thing in slight lengthy words so that it will be easier for you. The concentration of both carbon dioxide and hydrogen in the capillaries of metabolically active tissues is higher than that observed in the capillaries in the lungs, that you all know. In the tissues, carbon dioxide is converted by carbonic anhydride to carbonic acid, which spontaneously loses a proton, becoming a bicarbonate. That means now let's see, carbon dioxide is produced in the tissues. First of all, bind with water to form carbonic acid. This carbonic acid ionizes to form H plus and bicarbonate, and this about the bicarbonate will be there in the next class also, and that will it will also be neutralized by the chloride shift. And this hydrogen, there is the hydrogen is formed, and this hydrogen means decrease in pH, and this decrease in pH, presence of hydrogen means this hydrogen will bind with the negative charge of the hemoglobin molecule and with the ionic bond, and it will be able to form the salt bridges, and that salt bridges will stabilize the taut form, not the relaxed form. The taut form means deoxygenated form, and that means it will favor the deoxygenated form, means in the tissues oxygen will be released. The hydrogen produced by this pair of reactions contributes to lowering the pH. You all know this differential pH gradient favors the unloading of oxygen in the lung. Thus, the oxygen affinity of the hemoglobin molecule responds to small shift in pH between the lungs and the consuming tissues, making hemoglobin a more efficient transporter, especially because of the change in the pH. As we have said about the two conformations, one is the taut conformation and the other is the relaxed conformation, taut form and the relaxed form. And this taut form is favored when there is high concentration of hydrogen ion as occurs in the tissues. Taut form is favored means oxygen will be released. Okay. Now let's see this curve, the same oxygen dissociation curve. Now let's see, there are two curves for hemoglobin. One is for pH 7.2, the other one is for pH 7.6. Okay. What do you understand with these two curves? Just have a look. Now this at the lower pH means there is the right shift. Can you see the right shift? Lower pH, this is 7.2, this is 7.6. This is the lower pH, means higher hydrogen ion concentration. In the higher hydrogen ion concentration or lower pH, there is the right shift. There is the right shift. There is the right shift means to obtain the same percentage saturation with hemoglobin, the partial pressure of oxygen should be more. That means if the same partial pressure of oxygen is there, that means it has to release the oxygen bound with it. This is how the tissue and the cell gets oxygen. Okay, decrease in pH results in decreased oxygen affinity for hemoglobin and therefore a shift to the right in the oxygen dissociation curve. That means at lower pH, here you can see at lower pH a greater partial pressure of oxygen. At lower pH, greater partial pressure. This is 10, 20, 30. Okay, this is 20, isn't it? Now at 7.6 and for 50% saturation with oxygen, only partial pressure 30 is required. But for the same percentage saturation at pH 7.2, partial pressure 30 mmHg is required. That means in these conditions it can deliver. Higher concentration is required means in the lower concentration it will be able to, from the lower concentration it will be able to deliver the oxygen to the tissues. Bohr effect can be represented as hemoglobin oxygenated form plus hydrogen ion will be forming deoxyhemoglobin plus oxygen. That means with the increase in protons or lower partial pressure of oxygen shifts the equilibrium to the right. Shifts the equilibrium to the right means oxygen will be released. Where oxygen will be released? Where there is pH is low or the presence of S plus ion, concentration of hydrogen ion is more. Where it will happen? It will happen in the cells and tissues. And cells and tissues, that means this hydrogen ion will bind with the hemoglobin, and in that process this oxygen will be released so that it is able to deliver the oxygen which is bound from the lungs in the tissues. Now next molecule, effect of 2,3-bisphosphoglycerate on oxygen affinity. What is the role of 2,3-bisphosphoglycerate? This 2,3-bisphosphoglycerate also has the important role in delivering the oxygen to the tissues. 2,3-bisphosphoglycerate is an important regulator for the binding of oxygen to hemoglobin. It is the most abundant organic phosphate in the red blood cell, where its concentration is approximately that of hemoglobin. So high concentration, as you can see. And this 2,3-bisphosphoglycerate is found in Rappaport-Lubberin cycle. Rappaport-Lubberin cycle is just a side cycle which occurs from the glycolysis. In the glycolysis, there will be the formation of 1,3-bisphosphoglycerate, but this 2,3-bisphosphoglycerate will be found in the Rappaport-Lubberin cycle. 2,3-bisphosphoglycerate is synthesized from the intermediate in the glycolytic pathway. Binding of 2,3-bisphosphoglycerate, we cannot say the diphosphoglycerate that you all know, because this 2,3-bisphosphoglycerate phosphate groups is attached to the second and the third carbon, not on the same carbon. That's why we have to call it as bisphosphoglycerate, not diphosphoglycerate. 2,3-bisphosphoglycerate decreases the oxygen affinity of hemoglobin by binding to deoxyhemoglobin but not to oxyhemoglobin. That means it is bind to deoxyhemoglobin. That means it will bind to the hemoglobin and oxygen will be released at the same time, simultaneously. This preferential binding stabilizes the tau conformation of deoxyhemoglobin. That means it also helps to release the oxygen in the tissues. The effect of binding to 3-BPG can be represented as, you can see, same as the case earlier case, same as that of hydrogen. Oxyhemoglobin plus 2,3-BPG, hemoglobin bound with 2,3-BPG, that is the deoxyhemoglobin, and the oxygen is released. This is the condition in the tissues. Okay, binding. How this 2,3-BPG will bind, we'll see. One molecule of 2,3-BPG binds to a pocket formed by 2 beta globin chains in the center of deoxyhemoglobin tetramer. We'll see there the picture also. This pocket contains several positively charged amino acids. This 2,3-bisphosphoglycerate means it contains the phosphate group which is a negatively charged, and that has to be fixed with the positively charged amino acids. Now, pocket contains several positively charged amino acids that form ionic bonds with this negatively charged phosphate groups, this positively charged, especially lysine. A mutation in one of these residues can result in the hemoglobin variance with abnormally high oxygen affinity. If it is having the abnormally high oxygen affinity means it will be carrying the oxygen but not delivering to the tissues. 2,3-BPG is expelled on oxygenation of the hemoglobin. We'll see here. Okay, now this is the 2 alpha and this 2 beta, and this 2,3-BPG sits in a place just between the 2 beta chains. A single molecule of 2,3-BPG binds to a positively charged cavity formed by 2 beta globin chains of deoxyhemoglobin. That means in the deoxyhemoglobin form it will be stabilized. That means when it binds, oxygen will be, when it starts binding, oxygen will be released. Okay, now what is the significance of this 2,3-BPG, 2,3-bisphosphoglycerate? Okay, we'll see. Hypoxia, that means adaptation to high altitude, or in the case of obstructive pulmonary emphysema. In these conditions, just like in the case of high altitude, what happens? The concentration of oxygen is low, but our cells and tissues will be requiring the same amount of oxygen, and to deliver the oxygen to the tissues, that is the role of 2,3-BPG. If 2,3 more concentration of 2,3-BPG is synthesized, that means it will bind more, and if it binds more means more amount will be released in the tissues. Next is in the case of anemia. Case of anemia, you know, low red blood cells are there. If though low red blood cells are there, then it has to deliver the oxygen. Low red blood cells means low amount of hemoglobin that has to deliver the oxygen. And if 2,3-BPG levels are increased, what will happen? That will simply deliver whatever has been carried. And when coming to the lungs, it can again bind, and when going to the tissues, it will be delivering. So this 2,3-BPG binding with 2,3-BPG helps to deliver the oxygen to the tissues. This is most important in case of hypoxia, just like the adaptation to high altitude. Next one is in the case of anemia. Okay, there is one clinical significance in the case of blood transfusion. So, what happens is storage of blood in acid citrate dextrose medium results in decreased concentration of 2,3-BPG. So ionosine is added to the medium. What happens? If this ionosine is added, the ribose moiety of ionosine gets phosphorylated and enters into the HMP shunt and finally gets converted to 2,3-BPG. So ionosine is added. This is in the case of blood transfusion. Why this ionosine is added? To produce 2,3-BPG. Because during the storage, the concentration of 2,3-BPG will be lowered, and if 2,3-BPG is very much lower, then what will happen? Though the blood is transfused, that transfused blood will not able to deliver the oxygen to the tissues. That's why to increase the concentration of 2,3-BPG in blood transfusion, ionosine is added to the medium. Next is the case of fetal hemoglobin. That is, we are talking about the clinical significance of 2,3-BPG. The binding of 2,3-BPG to fetal hemoglobin is very weak. Binding is very weak means fetal hemoglobin will have more affinity to oxygen, and this is also important. Why this is important? Because of this hemoglobin F. This hemoglobin F has a higher affinity for oxygen compared to adult hemoglobin, and this is useful for the transfer of oxygen from maternal blood to the fetus. That means oxygen is having a high affinity to the fetal hemoglobin than that of the adult maternal hemoglobin, so that the oxygen can be easily transferred from the maternal hemoglobin to the fetal hemoglobin, and the baby inside the womb would be, fetus inside the womb would be able to get the oxygen. Now, in short, the role of chloride in oxygen transport. Chloride is bound more tightly to deoxyhemoglobin than to oxyhemoglobin. This also facilitates the release of oxygen in the tissues. Facilitating the release of oxygen in the tissues, that means causing the right shift of the oxygen dissociation curve are, here you can see the hydrogen ion concentration, 2,3-BPG, and this the chloride. All helps to release the oxygen in the tissues. Chloride is bound more tightly to deoxyhemoglobin than to oxyhemoglobin. This facilitates the release of oxygen in the tissues. Once produced in the red blood cells, bicarbonate freely moves out and equilibrates with the plasma. In order to maintain neutrality, chloride enters into the RBC and binds with the deoxyhemoglobin. This will also study as a chloride shift separately also. In order to maintain the neutrality, this chloride will enter into the RBC and bind with the deoxyhemoglobin. Chloride will enter into the RBC and bind with the hemoglobin, and in that process, oxygen will be released because it will favor to the deoxyhemoglobin. This also helps to release the hemoglobin to the tissues. This is about the oxygen dissociation curve. More things like how much percentage is carried and by which process, because even not only the oxygen is carried by the hemoglobin, but it is also involved in carrying the carbon dioxide from the tissues back to the lungs. And about the chloride shift we'll be studying in the next. Oxygen dissociation curve. Oxygen dissociation curve for hemoglobin and myoglobin are different. For hemoglobin, rectangular hyperbola or the hyperbolic shape, and for hemoglobin it is sigmoid shape. and this sigmoid shape of oxygen dissociation curve indicates the cooperative binding of oxygen to hemoglobin, and this is the simple binding of oxygen to the myofibre. This is all about the today's class. If you have any questions, you can just unmute your microphone and ask. Okay. In the meanwhile, I will download the attendance. This oxygen saturation for dissociation, oxygen dissociation curve is very important in examination point of view also. Go through it once, twice, or even more. If you have doubt, ask right now or later on also you can ask. If I need download it or not.
Sir, can you explain in the child one case?
Sorry. Hello.
In the child case. In the child case, sir.
Yeah. Oh.
Can you explain?
In the child case means they are having the fetal hemoglobin, and that fetal hemoglobin has low affinity with 2,3 BPG. Low affinity with 2,3 BPG means it is having the high affinity for binding with the oxygen, so that it can easily take up oxygen from the adult hemoglobin of the mother and deliver to the baby. As being the high affinity, it can easily take from the adult hemoglobin, which is having the low affinity for oxygen. But if you talk about the affinity for 2,3 BPG, it will be just the reverse.
Thank you, sir.
Okay. The binding of 2,3 BPG or to fetal hemoglobin is very weak, it's low affinity. Okay, any more questions? Any more question? Okay, no questions. This is the end of today's class. We'll continue with the hemoglobin, different types of hemoglobin in the next class, followed by hemoglobinopathies in the other class. Okay, thank you, class.
Thank you.